I suggest we get started only because I hope to have enough time to have a meaningful discussion about the action plan. And as you saw the agenda, by the time we get there, if we don't get started and do this all in a timely fashion, we won't have time to discuss the issue at hand. So first, let me thank in particular Masaki Takada, who has been spearheading the SLIT-J project at Sendai for organizing this and stimulating the organization of this discussion. It's hosted by Tohoku University, and it's a distinct honor to ask President Ono to make some opening remarks before we introduce each other and proceed with the agenda. So, President Ono, please.
Well, thank you very much, Jerry. Hello, can you hear me?
Yes.
Is audio okay? Good. So thank you for joining us today for our second World Summit of Synchrotron Radiation Facilities. We are again, after the first summit in Sendai last year, we are here together again to address COVID-19. Well, to overcome this pandemic requires collective efforts from all over the world. I am happy that the contributions from the synchrotron community can provide special opportunities to advance research on the new coronavirus. Along this line, Tohoku University also has put together a list of ways that synchrotron radiation-related technologies can be used for COVID-19 research. Today, in a limited time, we work towards adopting a new communique at the end of this meeting that will unite all synchrotron radiation and X-ray free electron facilities around the world in the global fight against the COVID-19 pandemic. We are all in this together. So thank you very much.
Thank you very much, President Ono. The next item on the agenda is the introduction of delegates. Proceed with the order that I distributed to all of you, beginning with, yes, welcome address, introduction of delegates, approval of agenda. Good. So I see here already Paul McIntyre, who's the director of SSRL, Mike Dunn from LCLS, Stephen Streifer from APS, Stephen Cavan from ALS, and John Hill from NSLS, and Joel Brock from CHESS, and joining us as well, the former director of ALS, a professor at the University of California, Berkeley Emeritus, but called back for service, and past president of the APS, Roger Falcone. I also see Francesco Sette, Director General of the SRF, Edgar Weckert, who's Research Director in Photon Science at DAISY, Robert Feidenhansel, who's the Chairman of the Board, European XFEL, Jean Daillon, Director of Soleil. I didn't see Andrew Harrison, perhaps he's here. Ian McNulty, welcome. Gabriel Epley isn't here, but Leonid Rifkin from PSI. Katerina Biscari is here from AOBA, and representing Elettrosynchrotron in Trieste is Claudio Massavecchio. Ishikawa Sensei is here from Riken, Andrew Peel from the Australian Synchrotron, Richard Garrett, who's an advisor at ANSTO, Guhui Lu from TPS, and Inso Ko from PAL, and some others. So if I've missed your name, please speak up. I see Sakura Pasquarelli, who's a research director at ESRF, and some others that I don't know. So, Martu? Yes?
May I speak? I'm sorry that you don't know me. I'm Jan.
Oh, Jan, I'm sorry. The trouble is I have many things on my screen, and you know, at my age, I can barely focus on one.
Don't worry, but I had to make the joke.
Yeah.
Thank you all for inviting me to join this group. I'm representing the BESI facility in Berlin, which is run by the Helmholtz Zentrum Berlin. We are the counterpart of the Petra facility that Edgar Weckert represents here. So like ALS, APS, we have Petra Bessi in Germany. Thank you very much.
Yes, thanks. And there's some other names on the list that I don't recognize.
Yes.
Perhaps I can speak up. I'd like to introduce from Max 4 my guest, Marjolein Tunissen. She's our Life Sciences Director and can speak knowledgeably about our program regarding COVID-19 when questions come up.
Mm-hmm. Thank you. Anyone else that I missed? Delphine?
Yeah.
Yeah.
Yes, Delphine, I'm the head of communication at ISRF.
Uh-huh.
May I just
Yes.
say that I was not missed, but I want to say that I'm also representing the LEAPS, the League of European Accelerator.
Oh, yes. Thank you, Katarina, please. I appreciate that very much. Okay, so now we move on to the next issue, which is approval of the agenda. So what, as you see before you, we've had the introduction. I've taken the liberty of introducing all of you, and next is an approval of the agenda. I hope that what we're going to do in the next 50 minutes or so will be successful. The next item would be highlights from facilities. Let me emphasize, many years ago, I learned that in particular in the structural biology community, there's a tremendous sensitivity to discussing things that people will do rather than things people have done. So when it comes to highlights, please discuss only those things that are generally available in the literature. Whilst they may only be in the form of a preprint on the archive, I think that's the sorts of things that we should share amongst ourselves. And then we will discuss the action plan, the draft of which is in front of you. If there are no objections, let's proceed to the highlights. Do I hear any objections? Everybody's nodding yes. Good. So I'm going to take the liberty of asking Stephen Streifer to speak for the DOE facilities. He co-chairs an advisory committee, a task force, I guess, but Stephen, please correct me, that is part of the DOE effort to address this issue. Stephen?
Yeah, thank you, Jerry, and thank you very much for the opportunity to talk a little bit about what's going on broadly within the Department of Energy and also at the Department of Energy synchrotrons. So very early on, the Department of Energy gathered together the 17 national labs that make up the Department of Energy complex, all of the labs that do make up the complex, to work together in a interlaboratory task force that the Department of Energy has named the National Virtual Biotechnology Laboratory. And this laboratory has been formed, this coordination board has been formed to try and tackle problems that are relevant for COVID-19, for which the Department of Energy has unique capabilities that can contribute to the international pandemic. So the areas right now that the working group has pursued are in epidemiological modeling, in clinical and testing, research and development, including development of new testing methodologies for the virus, in advanced manufacturing and supply chain issues. As everyone is aware, the supply chains are stretched very thin, particularly for personal protective equipment and for many supplies that are needed for testing. And finally, most relevant for this group, a thrust in medical countermeasures or medical therapeutics, which essentially consist of computational drug discovery associated with structural biology, where of course the US light sources are very key for doing that structural biology work. So there has been about $20 million of investment so far from the Department of Energy in the last month in these different areas. The US government has moved very quickly to try and support R&D. Within the light sources themselves, I won't say too much about this just in the interest of time, but all 5 of the Department of Energy light sources are currently operating for structural biology work on COVID-19. In addition, we're also pursuing other research projects related to COVID-19, for instance, looking at new materials for personal protective equipment that might have antiviral properties or that might be more easily sterilized for reuse in an environment where there simply isn't enough, for instance, filtering paste piece respirators to go around. Just very quickly, I do want to compliment the entire cohort of light sources around the world for contributing to the epidemic. What we're doing in structural biology is really essential to addressing the issue in terms of developing both antivirals and ultimately a vaccine. As of last week, the APS had counted that there were approximately 138 structures that have been deposited into the protein data bank. I very particularly want to compliment Diamond Light Source for having deposited any one of those. They very early on started to do fragment screening to try to identify drugs that would be useful in the pandemic. But other light sources around the world, including APS, the Shanghai Light Source, BASI, Petra 3, and others have all contributed structures to the protein data bank. And we believe that this will be essential as we move forward. Most of the work has been on non-structural proteins and the main protease, again, with binding to inhibitors and to receptors from the human body. And then, of course, quite a bit of work on the spike protein that is associated with entry into human cells. We hope that we'll be able to make a significant contribution to the epidemic. I think coupled with computational drug discovery, the work that's being done in the light sources is poised to shorten the time to therapies from what is typically perhaps as long as many years, hopefully to a few more months. Of course, as everyone is aware, there are drugs in human trials now, and I think this will be crucial in addressing the epidemic. So thank you.
Thank you very much, Stephen. Let me now move to Europe. But before I do that, Joel, do you want to add anything from Chess?
No, I think we're we have our normal capabilities for structural biology and also for the small angle scattering biological sectors, size exclusion chromatography, and so on. And we're actually just coming up out of a down and hope to be up and firing again. The beginning of this coincide just with the beginning of one of our downs, so we're just coming back. Hope to be back up in a few weeks.
So let me move to Europe. Francesco, I know that ESRF is not operating, but perhaps you have a perspective that would be of value to the community.
Yeah, and indeed, the ESRF is not operational, but nevertheless, we are at an advanced stage in our commissioning process, and we have recommissioned structural biology, bioimaging beamline, as well as our cryo-EM platform. There are all a series of activities on the pipeline that more or less reflect many of the aspects that Stephen Streifer had mentioned. However, I believe that in this context, it's better to give a more European global picture. And in fact, the synchrotron radiation and XFEL facilities together had already made a COVID-19 position paper, and the European Commission and the national agencies are strongly supporting research on COVID-19 related topics on which the light sources are directly involved, and the level of funding is very similar to the one that was mentioned for the US, and indeed includes a major effort also on computational aspect. And I believe that the most appropriate thing to do now is that Katerina, on behalf of all the European facility, perhaps gives more details that are, I think, more appropriate for getting the global picture.
Yes, thank you very much, Francesco. Caterina Biscari is the president of LEAPS, and I don't dare just tell you what LEAPS stands for because I'll make a mistake. So Caterina, please, it's a pleasure to ask you for some comments.
Thank you. LEAPS stands for League of European Accelerator-Based Photon Source. It is a consortium created a few years ago where there are 16 members. More or less half of them are represented here today, and includes 19 facilities. And of course, each of us is by one side working independently on with the user communities and also with industries to answers to the COVID-19 problems. But as Francesco has mentioned, we are preparing this position paper where what we have done is to summarize which are the capabilities of the whole community, which of course are capabilities which are shared with the other part of the world, so structural biology, imaging, scattering, circular dichroism, IR spectroscopy, soft ray transit absorption, fluorescent imaging, and these are all capabilities which they have not been all of them used up to now. Of course, structural biology is the one which has been more used, but is there also to face what is coming in the future. And we had in the discussion we have inside LIPS about how to deal with this situation. We are facing the possibility not only of collaborating in terms of what are our capabilities, but to share also what are the best practice in terms of operation, because it's not only the the fact that we have to face the scientific questions, but also how to operate our facilities. Many of us have been forced to shut down for some period, or some of us are now operating in a reduced mode, mainly for COVID-19 related experiments. But it means that in the next year, we are facing a situation in which there will be difficulties, of course, for traveling around for the users. Therefore, we are also working in how to organize together in terms of offering our capabilities in the best way with collaborating with the user community, all academic users and also industrial users. And I will not go into the details of the results that have been already obtained by LIPS facilities, because they are all here represented in BESI and also Electra, and I think they want to comment, they are best placed than I am. Thank you.
Caterina, thank you very much. From BESI, Mr. Luning, is there anything you would like to add?
I mean, we had the lucky situation that in January there was a measurement taken by the group from Lübeck on the structure of the protease, and so we published this on March 16, which was right the day this was published in Science by this research group that took the data at BESI, was published in Science in March 16, right the day that in Germany the shutdown started. So the synchrotron radiation-based research got really good publicity due to that. And this was followed by activity news on activity taken up at DESI, and we also followed at BESI. So like all other facilities, we run for COVID-19 specific research only. Thank you.
And Claudio Massivecchio from Elettra.
Hi, hello to everybody. So Elettra is running against the COVID. Actually, people can apply right now, I mean, since yesterday, for beam time, and the application will follow a fast track, so that will not go through the usual PRP committee. There is a special committee which will act immediately, and we have both Elettra and Fermi open to proposals. We got already one proposal for Fermi, measuring how a drug attaches to a ligand. In this particular case, how a drug like chloroquine attaches to the ACE2 protein, which is, I mean, you know that the spike protein attaches to the ACE2.
Thanks very much. Before I move to others from Europe and to Asia, let me just give the opportunity to my colleagues in the US if they wanted to add anything to Stephen's remarks. Mike, Paul, Stephen, if not, I don't see anybody.
Yeah, Jerry?
Yeah?
I might just add that we did set up for the light sources a portal where we collect all the structural biology resources in one place that people can see. That didn't exist before in the US. So for the light sources and the neutron sources, is now a single place to go to see those resources.
Thanks very much. Mike or Paul, is there anything you want to add before I go to the rest of Europe?
I guess I'd add that there's considerable coordination among the light sources in the US in terms of our plans for sort of future operations. We've been discussing, in fact, we were discussing that today. So I think we'll work together on this going forward quite closely.
Thanks.
Maybe Jay, I'll just add one thing.
Sure.
From the X-ray free electron laser perspective, I think we would dearly like to learn from our synchrotron colleagues, lessons of how to perform mail-in samples and remote users to be hosted in.
Could you mail in the user?
Yeah, mail in the user indeed. Where over this next year or so, I'm sure international travel will be extremely curtailed, and the degree to which we can work together and host locally users that would otherwise have traveled to each of our facilities and learn lessons in terms of mail-in samples and remote operation is something we're looking at extensively at the moment for LCLS. We issued our call for proposals yesterday alongside the Claudio's proposal. It happened to be on the same day for rapid access for COVID research at LCLS when we turn back on in the summer. And so we're trying to think through now how best to enable that without users physically present. So, a forum like this, will be incredibly important to help inform that.
Thanks very much, Mike. Anybody else in Europe who would like to make some comments about activities, ongoing highlights that you might want to mention before we go to Asia?
Perhaps I could speak for Max 4. As probably most of you know, the situation in Sweden is a little bit different than much of the rest of the world.
Yes.
Max 4 does continue to operate. We have 13 beamlines taking light, 10 accepting users, at least in principle. And although very few users are coming to Max 4, we're taking quite a few samples, particularly on our structural biology beamline. Marjolein, can you comment on our program regarding COVID-19?
Yes, so like many of you, we have a fast access program running right now, where we do, a fast track if proposals come in. It's a little bit wider than just COVID. It's for all medical research of very high priority, but of course centered around COVID-19. Because there has to be an understanding that many very important experiments for mankind cannot be performed right now. But of course, it's centered around COVID-19. We are gaining a lot of knowledge on remote and mail-in, and even beamlines that running experiments where you thought that mail-in would not be possible, like our RPES beamline, are now trying to do this. So there is a lot of creativity. There are a lot of ideas coming up.
Thank you very much. I know that Edgar.
Maybe we should just promote what's going on at Petra. Petra was actually only 1 week down, or a bit more, because we had a fast track scheme involved for COVID research, and since then we are actually screening for the drugs against the main protease of the SARS virus. We have a so-called broad library, which is a repurposing library that we screen, and we are through with this. So we have something like 6,000 data sets, 4,000 compounds screened, and 13 hits so far. That means 13 compounds that bind to the protease, which are now go through all those biochemical stuff to see really whether they can prevent the virus from replicating. What they now try to do is there are two other proteases which are important in viral replications, and these are the next one where they want to screen. So the one is the papan-like protease, and the other one is the human transmembrane serial protease. Other than that, there is some work on drug delivery issues, releasing drugs slowly that might for exact dosing. DAISY has provided compute power for epidemic studies to our national Robert Koch Institute. That's the institution that does advice to the government in these epidemics. The cryo-EMS of CSSB are running for COVID work. And people have now really came up for biological imaging of infected tissue work, so that we had lung tissue. At presently, actually, people have infected, I'm not sure whether it's from dead people or from a biopsy from lung tissue, where they want to really see how the virus actually interacts on a molecular scale in the human lung tissue. Flash is, since the COVID-19 crisis, in shutdown, but is doing now machine studies. That's all.
Thanks very much, Edgar. Good. So let's
maybe, yeah, Jerry?
Jean, please.
Yeah, just very briefly. So like others, we have established a fast track for COVID-19, but probably more significant is that we just got a project approved by the National Research Agency together with a virology lab nearby and the Sanofi pharmaceutical company to express the COVID-19 proteins, grow crystals in vivo, screen them, and determine the structure. That's it. And I have to apologize, but I will have to leave you in half an hour for another meeting.
Good. That's when we're stopping, I hope. That's the plan. Good.
Gary, maybe a few words from European Exfil, if you allow.
Yeah, sure, please.
Good. So from European Exfil, we are shut down because of the superconducting accelerator due to curfew. We decided to shut it down in order to prevent potential mechanical damages, but we are starting up again very soon, sometime here in May, and we are trying to organize it via a big community proposal to get all people in, and by that increase collaboration across different user groups, and also reach out to people who are not normally using synchrotron or X-ray variation. And we have opened our bio laboratories. We have a bio support laboratory, which is actually involved in the work going on in the Hamburg area.
Thanks very much, Robert. So, Ishikawa-sensei from Riken Spring 8 Center, can you begin the discussion about what's going on in Asia?
Hi. The Spring 8 is continuing operation even in this emergency to address the urgent challenges associated with COVID-19. We have been accepting the proposal of structure analysis of protein directly related to COVID-19 and other proposals, including from Tohoku University. And our group at the University of Tokyo is now conducting soft X-ray spectroscopic research of polymer materials, which are used for extracorporeal membrane oxygenation ECMO machine. Yeah, that prevent the blood to, that make the blood flow easier. Now we have learned that synchrotron radiation not only contribute direct to research related to COVID-19, but also many indirect contributions. And now we have very restricted people traffic. And we are only accepting the proposal of remote access or our staff.
Thank you very much,
Okay.
Tetsuya. Andrew Peel, is there any activity? Does the Australian synchrotron operate now?
Yes, I think, trying to think of what else I can add to the comments that have already been made. I think like many other facilities, we're open for COVID-related research. I guess one of the things I'd add to that is that the user community is so inventive that we've seen quite a few interesting proposals that claim to be COVID-19 related. So we also imposed a requirement that people be prepared to talk publicly to the media about the fact that their research is helping in the COVID-19 efforts, which made a bit of a step change in how many inquiries we were getting. I think the other thing I would mention is, like some others, there is increasingly some additional work that's not just structural biology related. For instance, we are doing some work in the coming week looking at virus detection strategies using infrared spectroscopy. And there's a number of other interesting proposals as well. And maybe two other points I might add as well. One is, I think we can't underestimate the importance of the message that gets out there, not just from doing the research, but the fact that we are doing the research. I think from one story that we'd had at the Australian Synchrotron relating to some work on some non-structural proteins, we got coverage on all of the national news television stations. And it's an incredibly important thing to support messaging to government about why they're investing in major pieces of infrastructure. So I actually posted a comment on the back of a Physics Today article that came out just recently to that effect as well. But it's certainly helped us with our discussions with our government. Then I guess the last thing, or one of the last things I'll mention is that I think we're also preparing for a different future over a longer period with limited user travel. I think Australia is particularly susceptible to that one. And looking at mail-in and other strategies, not just on structural biology, but on other beamlines as well, or increasingly on other beamlines as well. I guess another part is the link up between other organisations too. So I think it's already been mentioned, the link up between synchrotron facilities and neutron facilities. Getting that message out also has been really useful in terms of coordinating some of the efforts that are happening nationally.
Thank you very much. Professor Liu or Professor Chen, is there anything to add from NSRC in Taiwan?
Yeah, I would like to say something about TPS. Could you hear me?
Yes.
Okay. I think in Taiwan, we should, thanks to the excellent job done by CDC in Taiwan and the well-managed ball control. So currently, our machine, TRS and TPS, is running a schedule. We done it in last year. And but the border is controlled. So we actually send a message to our user. We are providing mailing service, and we are also testing some of the remote accessing program. And also, we already launched out a quick access program since mid-March that will help those program that's fighting for the COVID-19. I think currently there are several proposals that's already through this through spokesperson or the quick access program. There's some result, but most of the results were based on the SARS research result about 17 years ago, and they remodify those protein or some structure, try to find a good drug to cure the disease. And I think probably Junrong can speak more about those protein crystallography progress and how many proposals we already help the institution or the company solve during this period. Yeah, Junrong, probably, please.
Just a minute, please.
Okay.
Yeah.
Can you hear me? Okay.
Yes.
So as Director Low say, we have the TPS is running normally, and so the users carry out their project, I think, in the schedule. But we, because international users are banned because of government policy, so we cancel some international users. And so this is a beamtime we'll be using for the domestic user and mostly for protein crystallography and small angle scattering and structural biology study. So we in mid-March we call for the priority project for the COVID-19. So right now we received a few proposals, and then this is a quick access. So we had the rapid review processing, and then after that we arrange the bin time for the users. So
have there been any results?
Yes, yeah, and then hello.
Yeah, I'm asking, have there been any results? So time is of the essence. So if it's just accepted proposals, we're very excited that that's going on, but have there been results that have been put out in the literature on the archive?
Think they are getting the result, but not published yet. But they, I think they are getting published. Yeah.
Okay, so, thank you very much.
Okay.
So I think it's now time that we get to the meat of the discussion. And what I first recognize is that in this action plan, and let me thank in particular Stephen Streifer, Edgar Weckert, Francesco Setti, and who did I forget? Anyway, one or two others who have helped, Ishikawa Sensei, who have helped to develop this draft communique. What I find interesting is that many people have emphasized on the action plan, item 4, exchange and experience on users' remote access, sample mail-in procedures to maintain the experimental activity without travel. I think that that combined with number 3, the study of development of an IT system, so that we can exchange ideas of how to do this. And as several of you, Mike and others, have pointed out, the world has changed, and we're not only going to see the need for remote access and other means of access for research specifically focused on COVID-19, but more generally. For example, as the gentleman from Taiwan pointed out, the facilities are running perfectly, but nobody can come into the country. So that requires a new look at the way we access these facilities and how we carry out our research. So I now open the discussion for anyone who would like to comment on the action plan, positive, negative, otherwise. What would be best is there's an option somewhere to raise your hand, I think. And if that's true, there's a chat option on mine. I'm looking. Is there an option? Yes, there is. If you click the participants button on the bottom, you'll get a list of the participants, and then you can hit raise hand. If you do that, I'll call on you one at a time. So please, Francesco, since you've contributed heavily to this document, do you have anything you'd like to add or comment? Yeah, John. So, is there anything specific you would like to add or enhance on what we've
I raised my hand.
I see that. So did I.
Good. So I'm fine with the communique.
You are good. Does anybody else have anything they would like to add? Maybe Roger Falcone, who has experience managing, or as we like to say in the US, herding cats, or managing the American Physical Society, which is an analogous job. Roger, what do you think?
So what I was surprised about is we haven't heard, at least I may have missed it, discussions of work on the vaccine. And I was curious as to, that's so much in the political forefront. Is there anything that people could say about working with drug companies on vaccines?
Anybody want to comment?
Yeah, I'll say a few words about that. So,
all right, Stephen, good.
Yeah, there actually is quite a bit of work that's going on. I think several of the light sources have mentioned that a number of pharmaceutical companies are actually working at the light sources. Of course, one of the problems is a lot of that work is proprietary, and it's a little bit difficult to know exactly what's happening. The antivirals are easier in terms of trying to identify compounds that would actually be therapeutic. I think Edgar gave a very nice summary of the way that you can go about that. There is an increasing amount of structural work of proteins bound to human receptor and starting to look at the ways in which the virus replicates and penetrates into cells. So particularly as antibodies are isolated and as the epidemic goes on, we will see, I think, a shift from research on the antiviral compounds to research on vaccines. It's just a much more complicated topic, so it tends to come on the back end. But we, even at the APS, we are starting to see some of that shift.
Thanks very much, Stephen. Let's see. Edgar, you raised your hand.
Yeah, but I think others were earlier, like Andrew and Megan and Paul.
I'm sorry. Okay, yes. So, Paul McIntyre.
Yeah, thank you. Thanks, Jerry.
Yeah.
I think just to add to what Stephen said, there certainly is activity on vaccines. It is a lot of it is proprietary. We're seeing that not only at the light source, but also in my directorate, the cryo-electron microscopy facility is part of it, and they're obviously involved in this kind of work as well. The reason I raised my hand was just with respect to the question of mail-in samples and remote users. One thing that we've been discussing within SSRL is how applicable that kind of model of user engagement will be from one community to another or one set of techniques to another. Certainly the structural molecular biology community has a tradition of remote access use of our facilities, but there are other kinds of science that we want to start up in the near future where that is perhaps less common, and there will be some concerns about sort of keeping people's data and their results controlled prior to publication. So, how to navigate those waters, I think, is something that we'd be interested in learning about how other people have experienced it.
Thanks, Paul. Andrew Peel, I think, Andrew, you raised your hand before Mr. Veckert, and then Katerina, I'll get to you.
Okay.
Andrew.
So first up, yeah, I'll second or third those comments about work with industry. In fact, I think in the first few weeks, we had more work from industry looking at solutions than we did from academic researchers. But the thing I put my hand up for was, and maybe it's a bit of an elephant in the room, the comment in the action plan about global cooperation among facilities. There are quite a few facilities, obviously not in this meeting. Is it intended that, that these sorts of actions would be shared with the entire community?
So let me express personally that, and Takata Sensei, please correct me. This is an open collaboration, and the idea is not to limit it, but rather to be open. Those people that I forgot or misinformed, I take full responsibility. This is not a closed group. The idea is in the near term to focus on a crucial problem worldwide, but I think also in the longer term for us to share as facilities experiences, as both Paul and others have mentioned, for example, with regard to more remote access, if you'd like, for things other than structural biology. So I hope that others share my desire that this is completely open and by no means an exclusive club. Katerina.
Yes, I appreciate very much the draft as it is. I would like to see somewhere something saying, even if we say we are all international, to say something beyond frontiers, because this is a message that would be good to.
I'm sorry, could you repeat that again?
Beyond frontiers, that we are working all together and we don't have frontiers among us.
Yes.
It's a message for the politicians mainly.
Yes.
And then I want to mention about this development of not only remote access, but also mail-in. This is something that also we are discussing with the European Commission. Of course, there are proposals for future projects which are related with COVID, but also with the different way of working is coming. And they are very much in favor also of developing programs, but this is something that we could do all together on helping the remote access with the new software, new way of working that we should share. And also another thing that was mentioned is to upgrade the biosafety level of our facilities. Most of us have a biosafety level up to 2, but then there are some of the experiments that could not be done with this safety level, and then to develop, at least in some of the facilities, higher level of laboratory so that more experiments are allowed also. And another thing is also to mention, it was mentioned by some of you, the importance also of collaborating with other analytical facilities, like, of course, the cryo-electron microscopes, but also the neutrons and others that can be involved in this general collaboration.
Thank you very much, Katerina. Also, I don't know if you have seen, but Masaki Takata pioneered a university industry collaboration at Spring 8 in polymer science and soft matter. And in discussions I've had with people at SLAC, one of the aspects is trying to bring people who have problems that are related. For example, several of you mentioned experiments for medical equipment, if you'd like, rather than specifically structural biology. Many of those people may not be aware of the sorts of things that X-ray facilities can do in a unique way, whether it's tomographic studies, the studies of soft matter. Ishikawa Sensei mentioned polymer. Okay, Richard, Garrett.
Jerry.
Yeah, just a quick point on the participants. It's almost certain the developing world will have a much different experience from this pandemic than the developed world that we mostly represent. There's a couple of synchrotrons we could invite in. They probably have some unique issues like Thailand and Brazil. So just want to put that out there.
Good. Edgar.
I have two issues. First, to Roger's question, I know that there are vaccines already in clinical trials in Germany by companies. To my knowledge, they have not used any synchrotron so far. I would like to pose another issue to you guys, and that's maybe a bit tricky. I mean, there are some, and we have heard of it, some molecules that are targets for the virus in the replication of the virus or also in sticking to the cells. And most of us do probably public accessible libraries for screening, drug screening or fragment screening. And I fear, or there is, let's say there's a non-zero probability that we screen all the same stuff against the same proteins, and that would be a waste of resources. So would the facilities be willing to reveal what has already been done or what's planned to be done, so not to replicate results? I mean, if there's a hit, you might to confirm it at another place. But the phase space in terms of molecular combinations of certain complexes is so huge that we probably should coordinate ourselves. What's going to happen, started by Diamond Light Source, I'm not sure who actually was the first, and that's the same what we do. As soon as we have a hit, actually 1 or 2 days later, the coordinates are in the PDB, so that everyone knows that that has been done. But I'm not sure whether it would be also important to know all the negative results, because out of, I don't know, 6,000 or what we screened so far, we had only 13 hits. So, and you don't have to replicate that. And I would like to get your opinion whether you're the people that do the work. These are big teams in universities, so they might have different perspective on that, would be willing to share this information.
Thanks, Edgar. Francesco.
Yeah, I was reflecting on the comment that Katerina has done. Perhaps we should consider including a 5th point in our declaration, which I was trying to scribble, something of the kind, coordinate the X-ray science facilities with the effort carried out at other analytical facilities, and in particular, neutrons, electrons, and nuclear magnetic resonance. I think that for the general public we have an opportunity also to pass a message of the role of analytical facility in this context. Everybody knows that the scientists are at universities, research organizations, and so on and so forth, but they need the analytical facilities. So to put this fifth point, could even strengthen more the case of analytical facility in the public perception of their role.
Thanks very much. A very good point. Roger, your hand is still up.
Yeah, so I wanted to add one more thing. I'm sure this is happening globally, but in the US at least, we seem to have increased our national debt by 10% in the last month by the $2.5 trillion investments in response. And I am hearing that most of the needs of the light sources are logistical in nature, from what everyone is saying. Organizational, logistical, but are there needs? I know that in the US we are considering an infrastructure bill where there will be investments in science, and this will come as sort of a fifth legislation package that is happening in the summer. Are there needs for increased investments to further address the crisis, or is it really just a logistical and organizational set of issues that we're talking about?
That's a good point. Richard, did you want to say something else? Or did you not unhand your hand?
Is my hand still up? Sorry.
Yeah. And then Francesco's hand is still up?
I take it out.
Ah, okay. So does anyone else? I'm coming with the impression that there's broad agreement with the draft that we've circulated. And I think, Francesco, you raise a point, a good one, you and Katerina. Point 5 would be to coordinate the X-ray science facilities with other scientific tools, in particular neutrons, cryo-EM, NMR, and other analytic methods. Does that capture what you are thinking?
Yes, I just sent you an email with this line to
Okay.
you and to Masaki.
Good. All right. I'm looking at the hour, and whilst this would have been much more enjoyable with a nice conference banquet hosted by Tohoku University in Sendai, and perhaps next year we can do that, and in a year from then celebrate the opening of Slit J. Perhaps it's appropriate now. First, let me suggest that all of you individually, please send us comments on the draft so that we may, in the next just a few days, circulate this rather than a draft as the communique from our discussions. But also, I want to thank all of you for joining, and it's a pleasure now to ask for some closing remarks from Muramatsu Sensei, who's the director of the International Center for Synchrotron Radiation Innovation SMART from Tohoku University. Muramatsu Sensei.
Hi, thank you very much. So please, we deeply appreciate all the delegate participants through the Zoom and YouTube. This is the second summit of synchronization facilities in the world. So X-ray science facilities people produce the cooperative strategy across the whole facilities worldwide to work jointly to overcome the COVID-19 pandemic. In Japan side, as you see our website, we have summarized the technologies related to synchronization, and then we can promote various studies aimed at controlling infectious disease COVID-19 as entitled. So please show the, yes, we are summarize this promising application of synchronization to fight against COVID-19. So we have listed 3 aspects. One is applications to supporting next week, the technology for drug discovery. So we exemplify the structural biological analysis of the interaction between antibody drug that inhibits viral infection and viral surface factor, and the next utilization of synchronization facilities expected in drug delivery of pharmaceuticals. Second, the material development for life support and cure diagnostics and preventing infection. Next, third is a developed, standardized, the next generation utilization of synchronization facilities. Okay, so today we have succeeded in the hosting second SR20 summit, and we will collaborate all the challenging problems with SR facilities with others, so that finally we can overcome COVID-19 in the near future. See you again. Thank you.
Thank you very much, Muramatsu Sensei. So what I suggest is that we circulate with Francesco and Caterina's excellent suggestion of a 5th point, the draft again, for your comments and approval, so that we may circulate this more broadly as the communique from this very important discussion. Are there any comments or objections to that path forward? Okay, so especially to my friends in New York, Mr. Hill and Mr. Brock, who are either waking up very early or going to bed very late, I want to thank all of you. And on behalf of Masaki Takata, I welcome you all to Sendai when travel is possible to see the exciting developments of the SLIT-J facility. I visited not long ago and saw the beginnings of the civil construction for a very aggressive but very positive step towards another synchrotron radiation facility joining the family internationally. So thank you very much all, and particularly thank you to President Ono and colleagues from Tohoku for hosting and organizing this very important discussion.